Imagine you wake up tomorrow and someone says, "Good news, we can edit your DNA now." And your brain instantly goes, "Cool, so do I sit in a chair while a laser goes pew pew and my genome installs an update like Windows?" No, that is not what's happening. What's actually happening is simultaneously more real and more intense. Because the first FDA-approved CRISPR-Cas9 therapy is not a magic ray. It's a stem cell transplant workflow where your own blood stem cells get edited outside your body, then returned to you, like sending your engine to a machine shop instead of trying to rebuild it while driving at 120 kilometers per hour. And yes, this isn't future talk. The FDA approved it. Quick note, this is educational, not personal medical advice. If you or someone you love has a blood disorder, the real heroes are hematologists and transplant teams. Talk to them. Now, let me tell you the story of how bacteria invented a weapon, humans stole it, and a disease that has caused generations of pain met its match in a lab. Sickle cell disease is a genetic disorder where a mutation affects hemoglobin, the protein that carries oxygen. Normally, red blood cells are flexible and round. They glide through blood vessels smoothly. But in sickle cell disease, cells can become rigid and sickle-shaped. They clog blood flow, starve tissues of oxygen, and trigger episodes of severe pain and organ damage called vaso-occlusive crises. And this isn't rare in the once-in-a-century way. The FDA cites about 100,000 people in the U.S. living with sickle cell disease. Think of your bloodstream like Lagos traffic. When things flow, life is good. When a few vehicles turn sideways across the expressway, everybody suffers. Here's where it gets weird and beautiful. CRISPR didn't start as a human invention. It's part of how microbes defend themselves from viruses, like an immune memory system that stores mugshots of past invaders. CRISPR literally stands for Clustered Regularly Interspaced Short Palindromic Repeats. Yes, it sounds like a Hogwarts spell only biology students can pronounce. The Cas9 protein is the molecular scissors. A guide RNA is the GPS. It tells Cas9 exactly where to cut. And in 2020, the Nobel Prize in Chemistry went to Emmanuelle Charpentier and Jennifer Doudna for developing CRISPR-Cas9 as a gene editing tool. Bacteria said, "Here's our security system." Humans said, "Thanks, we'll take that, and also the instruction manual." When people hear gene editing, they picture in-body editing, like a tiny robot swimming through your veins, changing letters in DNA. But the first FDA-approved CRISPR therapy works ex vivo. Cells are taken out, edited in a controlled lab, and put back. It's less like patching software over Wi-Fi, and more like remove the hard drive, repair it on a bench with gloves and microscopes, then reinstall it. On December 8, 2023, the FDA approved Casgevy for sickle cell disease in patients 12 and older with recurrent vaso-occlusive crises. It's described as an autologous genome-edited hematopoietic stem cell gene therapy. Translation: your own stem cells, edited, returned. A true personalized biological repair. Let's walk the trail. Step 1. Prep and eligibility. This isn't walk in, walk out. Patients undergo screening, evaluation for transplant suitability, and infectious disease testing. Step 2. Collect stem cells. Doctors mobilize and collect the patient's CD35 positive blood stem cells through a process called apheresis. Step 3. The CRISPR edit. In the lab, those cells are edited ex vivo. A CRISPR-Cas9 ribonucleoprotein complex is delivered via electroporation. And here's the key move. CRISPR targets the erythroid-specific enhancer of the BCL11A gene. This reduces BCL11A activity in red cell lineage, which increases gamma globin and boosts fetal hemoglobin, HbF. Translation? We're not repairing the original hemoglobin directly. We're flipping a powerful switch so your body makes a different hemoglobin that behaves better under pressure. Less repair the old engine, more install a backup engine the body already knows how to run. Step 4. Conditioning, the hard part. Before the edited cells return, patients receive myeloablative conditioning, high-dose chemotherapy to clear bone marrow space so the edited cells can engraft. This stage can include careful monitoring, supportive therapy, and considerations like seizure prophylaxis with agents like busulfan. This is serious medicine, not a casual procedure. Step 5. Infusion, the return. Finally, the edited cells are infused intravenously as a one-time dose. They travel to the bone marrow, engraft, and begin producing healthy blood cells. In FDA-reported data, 44 patients were treated in the key sickle cell trial. Of 31 evaluable patients, 29, that's 93.5%, had no severe pain crises for at least 12 consecutive months. A New England Journal of Medicine report found elimination of crises in 97% of patients for 12 months or longer. That's the moment scientists stop saying interesting and start whispering, oh, we might actually be doing this. Later, Casgevy was also approved for transfusion-dependent beta thalassemia. Regulatory summaries reported 91.4% of subjects achieved transfusion independence for at least 12 months while maintaining hemoglobin above target levels. This isn't one disease getting lucky. This is a platform proving itself. Now, let's talk about the not a Marvel movie part. The label includes serious considerations: engraftment failure monitoring, delayed platelet recovery, hypersensitivity risks linked to cryopreservative components. Common severe reactions include mucositis, febrile neutropenia, and thrombocytopenia. This is why treatment happens at specialized centers, because the science is sharp and the clinical reality is heavy. CRISPR is powerful, but biology isn't perfectly obedient. The label warns about off-target genome editing risk. None was observed in evaluated cells, but the possibility cannot be ruled out, and long-term significance is still unknown. Regulators therefore require long-term follow-up, including 15-year observational safety studies to monitor risks such as malignancy and assess editing effects across diverse populations. So no, CRISPR is not copy-paste DNA with no consequences. It's more like we can edit with stunning precision, and we still respect the fact that nature hides Easter eggs and booby traps in the genome. Here's the elegance. The therapy boosts fetal hemoglobin, the version of hemoglobin you used before birth. Higher HbF reduces sickling effects by lowering the concentration of problematic hemoglobin in red cells, meaning the solution was partly inside you already, like an older, reliable tool stored quietly in the attic. Casgevy's edit targets the BCL11A enhancer, so HbF production rises. This approbation matters for 3 big reasons. First, a regulatory line was crossed. CRISPR is no longer just promising research, it is approved treatment. Second, ex vivo editing is a safer first step. Cells can be measured, verified, and quality controlled before infusion. Third, the playbook scales. If we can remove stem cells, edit them precisely, and return them safely, it opens doors. Blood disorders today, potentially many diseases tomorrow. But real talk, access, cost, and infrastructure and equitable delivery become the next battles. Science can be correct and still unreachable. That's a human problem we solve with policy, systems, and compassion. So here's the ending.
A microbe built a defense system to survive viruses. Humans turned it into a genetic scissors and won a Nobel Prize. And now, in hospitals with real patients, the FDA has approved a CRISPR therapy that edits your own stem cells outside your body, sends them back in, and for many patients, ends the pain crises that once defined their lives. Not a sci-fi chair, a lab bench, a transplant unit, a months-long quest, and a new chapter in medicine. If you want the next episode, we can go deeper into why BCL11A is the master switch, what off-target really means, and how scientists measure it, why in vivo editing is harder than ex vivo, and the ethical line between treating disease and editing humans. Like, subscribe, and comment. Do you think CRISPR's biggest challenge is science or access?